Can this slime mold grow forever? And I understand your question is in two parts here. One of them is related to the time span. What is the actual lifespan of this creature? And the other one is, of course, maximum size. I will have to make another video about the maximum size. A lot of people have been asking that, so I will make a video about that. But for this one, I really want to focus on the time element because this is fascinating. Now, the short answer is yes. It would just grow and continue doubling indefinitely in size as long as you give it enough food. Which of course the growth gets out of hand very quickly in that case. But the longer answer is very interesting. Physarum polycephalum, this slime mold, actually emerged quite a while ago in the 1960s and 70s as an excellent cellular model for aging, for cellular senescence, because its aging mechanisms are very similar to a lot of higher eukaryotes like human cells. And so for quite a while, scientists were growing Physarum polycephalum and not observing any signs of aging, which makes sense because there are a lot of microbes that you don't see aging in. Like if you have a bacterial culture, you will never see aging of the culture itself. It will continue propagating as normal because, the species would just end. If the whole bottle of cells would eventually age and die, then you would not have the species. So naturally, you don't see that in things like yeast and E. coli or any of these like microbes like this. So it was assumed for quite a while that Physarum did not really have a distinct aging mechanism. And then this thesis was put out. And it was pretty groundbreaking because it showed that Physarum does have senescence mechanisms. I'll show you the pictures that this person took. Okay, this is the picture that the author took as just sort of the baseline, healthy, young Physarum specimen. It looks great, right? And now his project was essentially just taking a small piece, the tiniest that he could, small piece of this specimen, putting it on a new petri dish and growing it until it took up the entire petri dish, and then taking a tiny piece of that specimen and putting it into a new petri dish and repeating this. He did this hundreds of times and grew just when you consider the aging of like this tiny piece, and then you grow that out so that it's aged quite a bit by the time it's filled the whole petri dish, and then you take the tiniest piece of that, you see this kind of like reverse dilution mechanism, and you can see how that would result in very rapid aging for a cell like this, but over hundreds of petri dishes. Now what he started to observe was he took these progressive pictures as he started seeing some of these aging symptoms pop up, and I'll show those to you. So here's the first one, just very minor changes here, and so I won't spend very long on this one, but you can see some of these like divots starting to form in the leading front, almost like there's some parts of it that are just not quite growing as quickly. And remember, it pushes its nuclei, all of its nuclei, to the front lines here, so its leading edge. And so maybe what we're seeing here is an accumulation of aged nuclei that are having a hard time keeping up, or mitochondria possibly that are having a hard time keeping up with the nuclei. I'm not sure, but anyway, this is what he saw, and then you can see here it's getting a little worse, it's getting a little more distinct. And then here, this is where it gets really bad. You see this kind of lacing effect sort of happening, where it's not this healthy mat of the cell spreading out like this. It's missing some chunks here, and this of course is very fragmented. It starts to look like fractals kind of, because it's it's a very inconsistent leading edge here. And now this is very late stage senescence for this cell. It's lost most of the attributes of a leading edge here. In fact, it looks like some of these are kind of leading the way, and most of it is falling behind. And finally, this is just the chunk that he took off of that last specimen, really having a hard time growing. It's not doing much at all. And then eventually the whole cell just lyses and dies. But this is very interesting, because the theory here, there's a lot of really interesting stuff. The reason this is such a cool model for human senescence is that we see a lot of the same aging mechanisms, like aging mitochondria, aging nuclear DNA. And when cells do start to age, especially in this, well, this is the lysed cell, but in the one right before this, you start to see something called polyploidy in the nuclei, which is where they're not haploid, where they have one copy of the genome. They're not diploid like they're supposed to be, where they have two copies of the genome. They're polyploid, which means they have just a whole range of number of copies, to the point where some of the nuclei had like an order of magnitude more copies, 10, 15 copies in addition, and I think even more actually in some cases, 15 copies of the genome, and they were substantially larger. Now, this is probably not a cause of aging, it's probably a symptom of aging. It's probably that they're accumulating these copies to try, and they actually also accumulated nucleoli, and I'll show you, hold on. Okay, here, so here we have the inside of the cell, endoplasmic reticulum right here. This is the nucleus. Inside here, this darker spot is called the nucleolus. It is the primary site for mRNA synthesis and ribosome synthesis, I believe. And so you can imagine this dark spot is actually where a lot of the work happens to initiate the process of synthesizing proteins. And again, Physarum is a master of synthesizing proteins, as I described in one of my other videos. So a lot of people don't know that cells can have multiple nucleoli. So if it needs to produce an enormous amount of protein, it can actually have several centers. Physarum does this, and it accumulates more copies of its genome. It accumulates more nucleoli as it ages, just to keep up with the protein production needs, and probably the declining functionality of mitochondria. They are having a hard time keeping up with this. Okay, so before we talk about the next part, you're probably wondering how some species of microbes avoid this damage accumulation within their entire population, to have our entire population just dying out around the same time. And this is a fascinating mechanism. This is from a paper that has nothing to do with Physarum polycephalum, but they're expressing this in, I think they're talking about yeast cells in this particular case, but this applies to most microbes and actually human cells, but I will tell you in a second why it doesn't matter so much. This is this asymmetric cell division. The asymmetric cell division is where you have this cell that has accumulated some damage. And when it divides, it has this intense mechanism to concentrate the damage to the nuclei, to the genome, I suppose I should say, and to the mitochondria. It accumulates the damage of all of these into one of the cells. And then the other cell, here, this cell takes the damage, and this cell has all of the healthy, brand new young components. Isn't that fascinating? And so here we see, in this diagram, we see what's happening. This cell always has a totally healthy daughter cell. And even this cell can have a totally healthy daughter cell until a lot of damage has accumulated. And we're getting lysing in this cell, but look how many healthy cells this cell can put out before it's completely dead. Now this is how the population can continue like this. And now in this diagram, they actually figured out a way to break the mechanism, and so they did see the entire population dying out around the same time, which was fascinating. But you can imagine why this is a problem for humans, because in humans, we just keep all of these cells inside our body. We keep all of these cells. We have, yes, a lot of good, young, healthy ones, but also we keep all of the very damaged ones. Also, there might not be such a great mechanism of damage asymmetry in the cell divisions, but this is how a lot of cells do it. And you can also imagine how Physarum does not actually have this capability, which is why it is an excellent model for senescence, for cellular senescence for human cells, because it retains all of the nuclei. It never actually divides. It retains all of the nuclei within its cell. Now, something I just said was a lie. Oh, okay, this is so cool. I'm running out of time, though. I'm gonna have to make a part 2, which I've tried so hard to avoid. I'm sorry. But okay, here we go. Okay, part 2. So I was just talking about how some cells have this mechanism of localizing damage that has accumulated to mitochondria, to the genome, in the nucleus, to one of the cells that they are producing when dividing. And the other cell that is produced does not have damage and retains all of the new young, undamaged components of the cell that were just made. And the other cell does retain all of the damaged components. And this continues to happen until the cell that keeps accumulating all of this damage, it eventually just dies. But look how many daughter cells it can make with almost no damage. before that point. And then this paper specifically was very interesting because they found a way to break that mechanism within cells that distributes the damage asymmetrically. And so they made it so this whole population of cells was dying around the same time because they all had roughly the same amount of damage accumulating in the mitochondria and the nuclei. Okay, I also mentioned how this usually applies to like yeast and bacterial cultures. This is how they avoid mass die off in their populations when all of the cells have all this accumulated damage. This is how that works for them. Now, it does not work like this for humans because humans retain all of these damaged cells, and there's probably not the same components and mechanism involved with damage asymmetry in human cells. And of course, even if there was, our organs and our tissues would still be made in large part out of the damaged cells. Look how many more damaged cells there are than young healthy cells. So even if we did have mechanisms to separate the damage, we would still have the majority of our organs are these aged damaged cells. And so this is one of the primary mechanisms of senescence in human cells as well. And Physarum polycephalum is a really interesting cellular model of senescence because it has something similar to this. Because the cell never truly divides, it does not have this damage accumulation in some parts of the cell and the young healthy cells here. So it accumulates all of these damaged nuclei and damaged mitochondria. But, okay, picking up back where the last video left off, I said that one of the things I just said was not true. One of the things was a lie. And let me get back to this groundbreaking thesis. So this thesis by Russell Poulter really was very groundbreaking in the 1960s because he was the one who discovered this and first described this this cellular aging mechanism in Physarum polycephalum. He did amazing work. He did a great job. It's a very long project and he put a lot of work into it. But Poulter missed something. And this is fascinating. Okay, I will show you. So again, he took all these pictures of Physarum at various stages of senescence. And here we have this one, and then this one as it's slowly starting to get worse and worse. It just looks damaged and aged. And then this one, and it just kind of continues to die off from there. But this method that he used of taking a small portion of each specimen and subculturing it in its own new petri dish has a flaw. And it does not actually replicate what happens in nature. And let me show you something. So as Physarum crawls around, it leaves behind this stuff. It's called slime tracks or slime trails, much like a snail or something. And when it does this, this is mostly comprised of proteins and polysaccharides. And it leaves this behind as for a couple of different reasons. It's antimicrobial, so it prevents mold and stuff from growing and overtaking Physarum in any place that it's been and any food that it's left behind. And it also acts as like a spatial memory, which is very cool because when the specimen encounters some of these slime tracks, it can actually tell that it's been to that area. And it has mechanisms to sense how old the slime tracks are and when it was last in that area, which is very cool. Because it can tell, okay, maybe there's new food here if these slime tracks are very old, but it'll prioritize the new areas of exploration before going back to these areas. And it just has the scales of priority depending on how recently it's been to that area. But okay, if you watch very carefully in these slime trails and you let a specimen of any age grow across the surface and leave behind these slime trails and then keep them growing, which is what Poulter did not do. Poulter just set aside the petri dishes. I'm not sure exactly how he got rid of them, but set aside the old petri dishes and moved on to the next. And then when the culture had run through that one, pulled off a tiny piece, put the rest of the culture to the side. Okay, if you keep that plate in optimal conditions and let the entire large specimen move out of the plate, here's what happens. Here's another picture so that I can show you exactly what's going on here. So you can see here, I'll zoom in here. Okay, you can see here these slime trails around where the specimen is pulled away, and now it's just this one tubule, and eventually even this tubule will recede. But do you see something here? What do you notice in this picture? Can you see it? these little blobs of yellow. It's not usually this aggressive. You saw in the other picture there was virtually no yellow, but in this one, little specks of yellow. I'll show you. This is a plate that I've made in my own lab, and we didn't have a specimen crawl across this, but we had a really good method. That has some problems. We had a really good method of rinsing the slime trails off and then redepositing them on a new dish. But just to orient you, okay, do you see anything in this dish? The answer is probably not, but let me zoom in for you. Look at that. I was giddy when I saw this, and let me show you what a different plate that was a little bit ahead of this one looks. Look at that. Look at that. Okay, zoomed in picture for you. Look at the tiny specimen. Okay, and here's the same plate just a few days later. Look at that. Do you know what that means? Oh my gosh. It means that when these large specimens, no matter the age, are leaving behind these trails, they are also leaving behind tiny little fragments of themselves. Just the tiniest little last pinch of specimen as it's pulling away from an area. And you can see them here. And notice how many there are in this picture. Now, most of these will probably not make it. At least, at least a large number of them will probably not make it. But what's happening here? Does this look familiar? This is a mechanism by which Physarum can embrace this element of asymmetric cell division. I mean, it's an element of cell division entirely on its own, actually, which is alien to Physarum. It's not supposed to divide, right? That's the whole thing. I've been saying this so much in all these videos, but this is a mechanism of cellular division in Physarum polycephalum. And it can leave behind these cells. Some of them may be old and damaged and ready to die. Others of them may be brand new daughter cells that have a concentration of all brand new undamaged mitochondria and nuclear genomes. Isn't that fascinating? And so this is what we see when Physarum leaves behind these daughter cells. Even if the specimen that left them behind is aging and is going through these mechanisms that Poulter described in his thesis, the daughter cells that leaves behind can spring up and as the new specimens of the area as these daughter cells. And also there's these very intense implications for, could this mechanism actually be broader than this? Could it be that this aging specimen is going around this area, leaving behind a lot of cells that are damaged and that are just going to die, but some of the cells that are these healthy daughter cells, those ones grow from that little speck into a larger specimen that's still largely undamaged. And can it fuse back with the older specimen to bring down its average age? And could this be like an intense network of this like aging system, anti-aging system, where they're constantly replenishing each other? Oh, this is so fascinating. That just the implications are mind-boggling. Anyway, but it is for this reason that Physarum polycephalum is considered a biologically immortal species. Even though it does display signs of senescence, it can overcome them through this mechanism. Isn't that fascinating? And also, what could that mean for humans? Wow, that's probably a little ahead of its time, but isn't that fascinating? Just the coolest thing. Oh, and one more thing I forgot to mention, a number. Just to give you scale of the scale at which the senescence occurs, for reference. If you had a sample of Physarum that was undamaged, just brand new young Physarum that was a kilogram in size, which is very large, don't get me wrong, it could grow, before displaying signs of senescence, it could grow to be the mass of about Earth's oceans.